Voilà, ben on est ravis d'être à nouveau dans l'amphithéâtre pour à nouveau une animation scientifique. On est heureux aujourd'hui d'accueillir le professeur Van Belkum. Tu m'arrêtes si je dis des bêtises, qui est professeur de l'université Erasmus de Rotterdam initialement. Si, tu me dis, hein, si je dis des bêtises,
mais c'est tout correct.
Qui est directeur recherche et développement chez Biomérieux. Et donc il est un Hollandais qui vit dans la région lyonnaise.
Oui, j'ai gagné autour de 10 kilos pendant les derniers 5 ans.
Qui par ailleurs est l'éditeur en chef de l'European Journal of Clinical Microbiology and Infectious Disease et qui, si mes comptes sont exacts mais ils ne sont peut-être pas à jour, a à son actif un petit peu plus de 800 publications scientifiques où il est auteur ou co-auteur. 500, pardon. Bon.
Ça suffit.
Et qui va nous faire le plaisir aujourd'hui de nous parler d'un topic qui est Modern Antimicrobial Susceptibility Testing. Alex, c'est à toi. Merci.
Merci. Si vous voulez intervenir pendant la présentation c'est bon pour moi, hein, pose des questions. Même si vous le posez en français, ça va aller. Mais pour les diapos, je vais parler en anglais. Parce que,
en hollandais
en hollandais, si vous préférez. Un petit peu d'allemand, on peut mélanger néerlandais, français, allemand. Mais je préfère l'anglais parce que ça augmente la vitesse un peu et ça sera un peu plus efficace, je pense. Donc, what I'll be talking about today is technology, and as Jean-Marc already told you, I used to be academic and I'm at a company right now, so I'll mix in a bit of advertisement for the company, I'll mix in a bit of economy, so it's going to be like the previous time I had my presentation here. It's going to be a bit of a mix for two, and I think it's also important for you as a really academic audience to be confronted with the industrial world from time to time, because over the past 9 years I really experienced that there are major differences between what you do and what DJ just told me. You're free to do whatever you want. We always have to think products and customers. That's the background, and then you may think that we always think money, which is absolutely not the case. Of course, there's a money dimension, but there's also a money dimension here. If you don't get your grants, you don't have your work. If we don't sell our products, we don't have our work. So there's always that balance between, let's say, working for the patient and maintaining the viability of the institute that you live in. And here you work directly for the patient, and your salary is being paid at the cost of the patient, so to say. And we get our salaries at the cost of the tools that we provide to you in order to take care of patients. So there's a lot of things that are important in that field. And if you look at infectious diseases, then we always think that we're very big and that there's a lot of things happening in the world, and that this thing with antimicrobial resistance is absolutely key in medical care. I think you should also realize that if you look at healthcare and global healthcare and the global healthcare industry, and you do that with a very sophisticated company, then infectious diseases are not that prominently visible in this overall prediction of what the key big developments are going to be in 2019. Voice applications. I don't think that we think about that every day, but when I got here, my taxi driver talked to his phone and he said, I want to go to La Timone, l'Hôpital de Timone, and you get immediate tracking of the route towards the hospital. Tracking patients and patient data in a vocal manner and using artificial intelligence in clinical settings is going to be increasingly important in communicating between patients and doctors, but also in communication between instruments that do diagnostics, whether that's cancer or infectious diseases, the doctors, and then their patients. So this is just an example of things that are considered very important by the big people in the industry, and of course healthcare data analytics is something that is considered to be spectacularly important, and I'm sure that you know all about that because you do a lot of sequencing in the lab here, and generating big data and translating that into something that is helpful for a patient is not always as simple as we initially thought it would be. But still, microbiology is our business. So if you look at that, then there is traditional and conventional microbiology, and maybe that's not exactly the case for the laboratory here, but 75% of all the diagnostics that are being done is classical stuff. Stuff developed by Pasteur and Koch in the 1800s, and that's still maintained. I'm not going to ask you, Michel, how many petri dishes with artificial media you run through the lab on an annual basis, but if you do 2 million specimens a year, I'm pretty sure you do at least 500,000 classical cultures on petri dishes on an annual basis. So this is still very big and very important, and also important to remember. Consumables, absolutely key. There's a large fraction of the overall microbiology budget used on a global scale that is in consumables. And the market is considered to grow with about 25% between 2016 and 2021. And much of that is obviously driven by the resistance issue. So this covers not only the laboratory. There's a lot of other parties involved in our business: hospital pharmacists, clinicians, obviously patients, but also healthcare systems, governmental support, and what have you. Public authorities defining the way that the money is going to be spent, and in that same context, the laboratory is becoming increasingly complicated. It was already pretty complicated with a lot of manual activities and passing petri dishes from one person to another, and correlating that with serological data, and thinking about the impact of all of that in a sort of a consensus manner on your patients is important and complicated. But I already told you, and obviously you know, there's a big issue with multi-drug resistant organisms and the increasing prevalence of drug resistant organisms, especially in those regions where you would not, where let's say antibiotic usage and availability is always compromised. There is cost pressure in this age and this time and age of trying to reduce the medical costs on a global scale. Laboratory costs are obviously a target because people consider us expensive. I don't know whether people sufficiently realize that with that expense there comes a lot of expertise and a lot of good for the patients. But then again, there's always talks about money, and money saved in the clinical microbiology laboratory, or activities not done in the clinical microbiology laboratory, may save a lot of money in different departments within the hospital. So there's always that discussion on budget pillars between different departments that is confounding the costs and the cost savings that you can let's say impose in your own environment. Obviously, and something that concerns us all, rapidity is important. Nobody wants to sit and wait for 16 hours for an antibiogram to return from the laboratory. People want that in 15 minutes. People go to their general practitioner with a cough, and they want to know what's it caused by, and if it's caused by a bacterial agent, can I use this and this antibiotic, because you can tell me whether that bug is still susceptible to that antibiotic. And this is I also think, if I look at my daily job, this is the core aspect of what we're currently developing. Can you translate classical microbiology by using next generation sequencing into something that is more intuitive and more rapid, and which would not increase the cost by a factor of 10? These are the type of, let's say, research-based questions that we have continuously in mind. And of course, this is all to help improve patient care. And I'm sure you've heard of and you know the Mérieux family. They're pretty rich, I have to admit, but they're not driven by profits. It's still a company business, and working there is different than working in a pharma industry where people just have to, where people are just developing 4 or 5 molecules, and where you have to decide which one to pick and which team to fire and which team to maintain. So this is different, and we're more of a, I won't say a family, but something close to a family, and working together to work towards all this. So I'll be talking about laboratory business, and again, I'm sure you've seen all of this. If you look at the flow of microbiological diagnostics, then most of us concern the analytical part. Bacterial and viral identification, detection and identification, and downstream of that, bacterial and viral characterization, resistance measurements, virulence assessments, vaccine coverage, things like that. So this is sort of the core business, and because of that core business and its main impact financially and also on patients, these are things that are frequently underexposed. And again, today I will not talk about sample collection and the pre-analytical phases, getting the sample from a patient into a clean container, getting it from the patient to the laboratory, getting it from the laboratory acceptance structure into a diagnostic test. This is all very important in the phase before this analysis. And if you put a lot of effort in making this cheaper and faster, and you don't spend a bit of time optimizing this, then if you have a sample collection and pre-analytical and transportation phase that takes 4 or 5 hours, then why reduce the time for antimicrobial susceptibility testing from 6 to 3 hours? You gain 3 hours, but here you should be able to gain at least 4 or 5 hours. So rapidity is defined by the overall process, although people tend to focus on the rapidity in this part of the process only. And the same is true for the post-analytical part. If you generate a result at 5 minutes past 5 in the afternoon, when the lab is just 5 minutes closed, then you essentially lose 16 hours before the lab reopens and the result is communicated to your clinician customer and to the patient. So these are extremely important points, and I don't know whether you're 24/7 here. Fantastic. I think you're one of 10 labs in France that are 24/7. Maybe 20. Anyway, so this is still not commonplace, and if you want to improve the quality of your services, then these are the type of things that you need to think of. Getting the data from the lab to the treating clinician and then translate it into action for your patients is absolutely key. And this is what it's all about, mostly for people with bacterial or fungal infectious diseases. To me, this is the sort of the 7R, the R7 definition on what to do when antibiotic treatment is involved. An antibiotic should be given to the right patient, in the right species, so to say, right dose. And I'm sure you've seen it before. But the entire process developed and maintained in the clinical lab is ultimately aimed at this. And all of the types of diagnostics that help you identify susceptibility of a bug, the formulation, the need of a certain drug, the pathogen in question, these are all things that define the way that a patient is in the end being treated. And then antibiotics, beside the fact that they're essential for the treatment of patients with infectious disease, bacterial, fungal infectious diseases, they have their problems of their own. Antibiotics are everywhere, but resistance to antibiotics is everywhere as well. And I just put on 2 examples. The literature is absolutely staggered with this type of information. But if you look at, for instance, the antibiotic use on a global scale, then you can see that certain antibiotics in certain regions are much often or much less often used. So there is no uniform global consensus on which antibiotics to use in which way for which patients. Message number 1. The other thing is, if you look at the work from Frank Arestrup, published in Science a couple of months ago, you will see that you can find resistance in all continents, and that you can find it not only in your patients, but also in sewers, in the environment. So antibiotics are everywhere, resistance is everywhere, and that is severely complicating the work of clinicians. The other thing, and that's in correlation with the message of the previous slides, if you combine drugs and bugs, resistance is going to develop. And the use of antimicrobials, of antibiotics, drives that resistance level up. So there's selection, there may be even promotion of certain selective processes, but in the end, the more meropenem you use in a certain hospital, the more likely it's going to be that over time, and depending on the dosing, you'll find more resistance in your setting. So antibiotics are there, antibiotic resistance is pandemic, and the use of antibiotics induces resistance. So the message there is that you should be careful with your antibiotics, that you should use the seven right antibiotics for the correct group of patients, and that you should always perform surveillance, depending on whether you have the budget for that, for the emergence of resistance, even using your clinical data on assessing, keeping your finger on the pulse on what's happening in your region, in your hospital, maybe even in your country or internationally, is absolutely key. And that again is underscored by the fact that if you're infected with a drug-sensitive organism or a drug-resistant organism, there's differences in budget. Treating a patient who is infected with a resistant organism is more expensive than treating a patient infected with a susceptible bug, and the mortality rates are elevated. In this case, for the MRSA, MSSA example, that may be twice the mortality. So again, antibiotic resistance and clinical care are very closely integrated and interactive, and has effect on budgets and on patient welfare and management. So what do we have tool-wise to intervene with that and to make our world a little less complicated than it seems to be? Well, there's tools, technical tools, and that's what I will be discussing for the rest of this presentation. Antimicrobial susceptibility testing essentially defines whether a bug is resistant or not, whether you can use certain antibiotics or not, and doing this for an entire scala of clinical isolates that you obtain in your laboratories. That will also give you a means of defining the prevalence of certain resistance factors, defining the presence of multi-drug resistant organisms, and maybe even pan-drug resistant organisms that are not amenable to standard treatment anymore. So this, to me, is a very simple definition. AST is a microbiological procedure developed since the early 1900s that determines the concentration of antibiotic required to inhibit the growth or kill a microorganism. And we've got currently two major categories of these technologies. Either we have the growth-based, commonly referred to as phenotypic methods, and on a more surrogate level, more indirectly, molecular means. And then of course the omics technologies become more and more prominent, and we'll discuss that when the presentation proceeds. Using these technologies, the first question obviously remains, is the infection that a patient is presenting with, based on clinical assessment, on clinical symptoms, is that a viral or a bacterial infection? And for this, phenotypic technologies are not that useful. You need at a medical visit, you need to quickly inform or at least extract information from your patient and try to figure out whether this patient has a viral or non-viral infection. And for that, there are simple antigenic tests for group A strep or for flu A, but these are not the phenotypic tests. So these are mostly genotypic tests that fall under the category of PCR testing. And still, there are some debates on whether this is fully covered at the moment. I think that at the level of the general practitioner, absolutely not, because although there are many let's say molecular technologies that can be performed in small equipment that are point of care like, which can be used in the general practitioner's office, it's not commonly done. And I know that for instance the group A strep test has been made available for free to French general practitioners, and the use has been a big debacle, as they said in France. Apparently there's not only technical inhibitions, but there are also sort of philosophical barriers where people do not want to use certain tests because of, I don't know what reason, maybe education, maybe the way they're working, maybe the way that colleagues look upon these tests, or maybe even cost aspects, even in this case, there were no real impacts on that level. So here, molecular technology provides the opportunity to reliably distinguish between viruses and bacteria.
Probably there is one exception in this, which is, I tried to develop that with the failure with Biomayo. There is one exception was gynecologists. Because in here, as in many countries, the gynecologists were doing this. So they were picking up, for vaginosis or sexually transmitted disease, they were doing that. Even 20 years ago, they got a microscope to see trichomonas by themselves. And now, you know, it is a very, very big product in the USA as a doctor test to get these, you know, bands that you test for chlamydia, trichomonas, and so on. So there is one exception that I don't know how much it is now in the young generation, but in the olders, the only people that were having as a doctor a microscope were the gynecologists.
I agree. The only let's say softening factor there is that for most of the sexually transmitted diseases, if you have your result a day later, it's not going to affect the disease that much, or the suffering or the mortality or morbidity of your patient. But yeah, of course you're correcting that. So this is molecular technology, and there's a wide variety of tests available. And again, I'm going to just mention one example, which is a multiplex PCR syndrome test, with which you can relatively reliably distinguish viral infection from bacterial infection. The multiplex test contains like 15 viruses, a handful of bacteria. So doing that is a process that takes a swab and 45 minutes of your time. I don't know whether that's sufficiently short for patients to be willing to wait in a doctor's waiting room for the result, but at least it is point of care and it is quick. It's bloody expensive, so that prohibits broad application. But it shows that depending on whether you successfully distinguish bacteria versus viruses, this has a real impact on, for instance, the length of hospital stay. If you do your testing with this machine for identifying viruses or bacteria, and you look at the control group and length of hospital stay, you see a significant differences. So these tests, whether they're 100% sensitive or 100% specific is not known, but they do have a clear clinical impact, and this is important to realize. And then again, I just mentioned, let's say, our technology, but let me reassure you that this is a very dynamic domain. There are many companies now that have developed and sell and promote this type of syndrome-oriented multiplex testing, and this is going to be a very competitive market. And where I said that the BioFire test is relatively expensive, I think this market is going to be different over the years to come, with more competition and changes in price levels. And that's why you don't have to read this slide, but it just gives you an idea of what's around. And the other thing that's very important to realize is that next to PCR, we're now slowly moving in the era of next generation sequencing. I think you as a group a bit quicker than the average laboratory in France, in Europe, or on a global scale, but you need to realize that we now do PCR, and this seems like the ultimate solution. If you can do a complete sequence analysis of a clinical specimen with all the data, whether it's viral, bacterial, or even host genome and transcriptome related within half an hour, and technologies that can do that are on the verge of being developed, then PCR is going to be looked upon differently, maybe, when time goes on. There is still, the genomics arena is there. There are no genomic tests, except for cystic fibrosis in humans, that have been validated by the governing agencies like the FDA for diagnostic use. So we have to wait clinical studies, and sequencing everything in a clinical specimen can also result in getting too much confusing information rather than clinically relevant information only. So we'll have to see how this PCR and genomic tendencies converge, and what in the end will be the best will be the best approach for this type of diagnostic. I got friends in Labor Berlin in Germany, where they look at complicated patients only with genomics technology, and that might be a good approach. You can do your screening for the average patients with relatively cheap and non-complicated PCR-like technologies, and if you do not get the answers that you want to have, You can take it one step higher and use hardcore sequencing technologies to try and figure out something. And examples of successful diagnostics using that sort of approaches are slowly emerging. There are many papers already that describe cases where this type of sequencing really helped cure patients in the end. So that's the molecular part, and that's where you decide whether or not the patient is infected by a virus or not. And if you know there's microbiology, there's microbes involved rather than viruses, you enter the laboratory phase with culture and the entire analytical phase, and you start after your cultures or using DNA extracted from clinical specimens to look for the presence of resistance markers. I'll be putting in a little bit of a promotion for the phenotyping, because this is classical, this is well accepted on a global scale. Growth-based antimicrobial susceptibility testing is still the key method. There is a push towards molecular, but microbiological testing, viability testing of real bacteria is still something that is let's say at the core of our business. And if you look at some of these microbiological issues that sort of circle around antimicrobial susceptibility testing, you'll see that there's problems like slow growth. There may be small numbers of cells in clinical specimens. Bacteria are very easily scared, and then they won't grow anymore. They take like 3 or 4 hours to sort of revitalize, and things like that compromise this antimicrobial susceptibility testing. So you have to keep that in mind. These are limitations which prolong the turnaround time of antimicrobial susceptibility testing, but still you're working with the infectious agents, and you can look at the effect of antimicrobials directly on these living cells. And with genomic or with PCR-mediated technologies, this is more indirect. You're not looking at viable organisms anymore, and you may be looking at DNA from dead bacteria, for instance. So these are things that are a bit conflicting, and this is also why molecular testing has not replaced phenotypic testing completely. And in addition, the presence of a gene in a clinical specimen is not always conclusive, in the sense that the gene may not be expressed. If you look at vancomycin resistance, for instance, if the gene is in an Enterococcus, then you should be careful. If it's in another species, it may just be normal flora. So there's a couple of things that you need to think at, and for that, giving these problems and given the fact that the molecular competition is not like pushing the classical AST out of the lab, there's still a lot of effort going on to improve this technology and try to make it more sensitive, more specific, and particularly faster. Because now I think that's sort of the common feeling. An antimicrobial susceptibility test after a positive culture is at least going to take an overnight incubation, so like 16 hours. Sometimes you can do some things a bit quicker, but then in most cases you still have to wait for a complete analysis with multiple antibiotics and multiple bug concentrations to be finished. So some results can be obtained relatively quick. Getting the full panel of antibiotics for that particular organism may take significantly more time. So there is a need for AD and IST at the same time, and I'm not going to address that with one additional sentence. Mass spectrometry, which you'll be using in your lab here very, very frequently, has replaced the old-fashioned biochemical identification. So acceleration of identification has already taken place. And I don't see anything else but MALDI-TOF, MS, or maybe another mass spectrometry taking that place over the coming 5 years. Not even sequencing. Mass spec right now takes 20 minutes post-culture and like 20 cents of reagents. So it's very, very difficult to be comparative with another technology. So we really need to push the rapidity of antimicrobial susceptibility testing, and also try to avoid all of the problems that we're currently seeing. So what do we have? Well, I don't know whether this is visible. In the 1930s, Alexander Fleming saw inhibition zones, and then you got your resistance developing over the following 2-3 decades. And then agar dilution tests were introduced, this diffusion, macrobroth dilution, and all of the equipment that you're familiar with, because it's run downstairs here or upstairs, I don't know, in the laboratory. So nothing new there. And these are well accepted technologies, like I already mentioned before, and are supported by vast numbers of scientific and clinical publications that support the usefulness of this technology. The thing, what is happening right now, and that has been becoming more and more frequent over the past 10 years, is invasive technology. A lot of smaller companies say, well, these are the big ones, and we really have something that is more competitive. We can do things in like 10, 15 minutes. Really innovation-driven antimicrobial susceptibility testing. And this is absolutely lacking in the big companies, in the big technologies. And I say absolutely lacking is a bit biased because of my own, let's say, observation from nearby at a company like Genome. But it's also very difficult to innovate these technologies. Because I take Vitek 2 as an example, which is a machine, and it has a card, and you throw the card in, and it does exactly what you want to do. If you kick the machine off the stairs, you can pick it up, you set it on the table, and it continues working. It's very robust. There's huge databases with the clinical relevance of all of the data. There's guidelines for interpreting your MICs. So this is very solid, and it's been validated by the organizations like Marquage Européen or the FDA, and anything that you do in a system like that has major impacts because it needs to be re-evaluated by the FDA, which is costly. You need to do clinical studies, which take time and which take a lot of effort. And the other thing is we have an installed base of tens of thousands of machines. And Michel is not going to buy another machine because we painted it yellow or something. So you know, this innovation at this level is very complicated because you're sort of limited by all of the requirements, legal requirements, clinical requirements. But still, we try to push this a little bit forward because you have to, given all of the problematics that I showed you in the first 4 or 5 slides. Yeah, you have to innovate, and this is something that we call innovation. We have the Vitek 2 card, which contains 64 wells, and now we have a Vitek 2 card that contains 108 or 106 wells. I see you're all pretty much impressed with this. It's like very simple. So yeah, you have to find somebody who sees the relativity of all of this. So it's not like Nobel Prize winning type of innovation, but this is the type of innovation that increases the productivity of the equipment, which is valuable because you can run more antibiotics or more antibiotic concentrations in the same time frame as when you did 64 in the past. So it's certainly valuable. And for us, it's also relatively secure. You don't, of course, you have to show that this works nicely, but you don't have to do massive, very expensive studies with the FDA. And for us, the most important part is that this card fits in the machine that's on his desk downstairs and already does what it has to do for 6 or 7 years. So this is the way that you should think about, let's say, evolution in these big tools, and then we add some tools that will help you make your suspensions. We play with databases, but it's absolutely not at the level at which these smaller companies play. We do densitometry, which is classical, which has already been done by Fleming from the time onwards. Broth microdilution was done in tubes. We do broth microdilution essentially in a little plastic cassette. So this is technology that is as old as antimicrobial susceptibility testing. But right now, people are using isothermal microcalorimetrics in machines that are this big and that are capable of differentiating 1,000th of a degree centigrade of heat produced while bacteria are dying or surviving in the presence or absence of bacteria. So this type of technology, nuclear magnetic resonance, requiring equipment that feels like part of this stage, but you can use it to look at metabolic differences in the culture medium above your cells and see whether there's still metabolism going on in these cells. So are these cells metabolizing in the presence of an antibiotic? Then they survive, non-resistant. If not, they die, and the NMR technology tells you that you have a susceptible organism. And for all of these technologies, there are examples of publications and documents and patents that were reflected in my previous slide with all of these red arrows. For all of these technologies, there are now spin-off companies that are trying to develop this into a competitive and clinically useful technology. And they all claim rapidity, high throughput, low cost, rapidity, rapidity, rapidity. And then you have companies like Bruker who say that this equipment that has been pushed into the lab and is called mass spectrometry can also be used for susceptibility testing. And of course you can. And a lot of intelligent people have been developing like 6 or 7 different applications already. None of these technologies has made it into a routine assay yet, although Bruker is selling a test that they call Astra, if I'm correct, with which you can determine the disintegration or the destruction of beta-lactam antibiotics. And that's an assay that is supposed to be useful. I have a specific personal opinion on that, but I will not bother you with it today. And then this rapidity thing. All of these companies claim things and they demonstrate things with 1 or 2 bugs and 1 or 2 antibiotics, and then they say, yeah, it's going to work, but forgetting that there are more complicated bug-drug combinations that take more time to develop. But there's one in Sweden, in Uppsala, the company named Astrego, who did a little bit more work for one organism and about 10 different antibiotics. And they show that with their technology, they can define, let's say, a resistant susceptibility level within 15 to 30 minutes. And they do a very simple assay. They have a capillary system here, all different capillaries, and you can load these capillaries with bacterial cells, and you see them sitting here. And then you can, let's say, flux these different capillaries with different solutions. So you can take growth medium, you can take growth medium with a little bit of antibiotics, with a lot of antibiotics, and then you just follow the growth of these bacteria. And they have fantastic, you should go and visit their website. They got fantastic videos where you can just see the replication of bacteria within these little tubes, and in the end they just fill up these tubes. So they're pushed in from here at the beginning of the assay. They end up here because there's a sort of a capturing device in the microfluidic system here, and you can just see them grow to the outside, to the right side of these tubes. And this is fantastic. They have a little box which does it. They have a little disposable that contains these capillaries, and they're currently trying to push this into still a limited market, because they looked at E. coli and they're looking at something like a UTI, urinary tract infection type of assay, where you have 4 or 5 main pathogens, and where you have not too many different antibiotics that are clinically relevant. And this is a highly adaptable system. Essentially, the liquids that you use to flush here and to see whether or not multiplication is blocked or not can be modulated very easily. You can go from one antibiotic to another by just changing fluids. So this is spectacular. And again, like with the molecular stuff, they're not alone in this field. I think they're one of the more advanced companies. They have equipment, they have disposables, they have protocols. So this is going to happen. But there are many other companies that use all sorts of technologies like Raman, spectroscopy, multi-channel assays like Astrego does as well. So there's again, where I showed these invasive technologies, these are the companies that are trying to push that towards the market. And they have to be very persistent and strong in order to achieve a good market position. I'll speed up a little bit, because the other thing, and that's more of a business perspective, so I'll be very superficial with the next 4 or 5 slides. If you are Astrego and you have a new technology, most of these companies, they do not realize the effect of having something successfully on the market. You have to have a customer support and a customer reply function. If something goes wrong, if a bug emerges that doesn't fit your software or your databases or the equipment, you have to do something about it. And this is absolutely something companies do not like. And we have regular problems with our equipment and with our testing issues because customers observe things that seem to be, yeah, he's shaking his head, observe things that are not correct, and then you have to react. So if we have false susceptible E. coli for TCP, then that the FDA tells to all our customers that they should use a second method to verify any data that they consider useful in their clinical setting. So this is costly, and this is not something that customers like, so you need to be very reactive. And this was a problem that we could relatively easily solve from a phenotypic perspective and from a, let's say, from a user's perspective, and I will not even discuss that, but we at least had the opportunity to dig into that a little bit. And we were able to show that this had to do with hetero resistance, small subpopulations in our customer strains that sort of confounded the analysis. And is this something that can be done by a small and medium-sized enterprise? If Astrego launches their product and their customers say you have a problem with TCP, what can they do? It can be very painful to the extent that they may go bankrupt if this sort of thing cannot be solved. So having this sort of backup structure is something that many people seem to ignore. Well, we looked into the problem a little more intensely. We looked at the strains that showed this heterogeneity against TCP. We selected resistant subpopulations, and of course, like everybody does today, we did some genome sequencing. And what we were able to identify was a genomic fragment of about 10 kilobases that contained BLATM1. And BLATM1 was the only beta-lactamase in these strains, the single one. So we specifically looked at the activity of BLATM1 at the protein, at the transcription, and at the genome level for all of these strains. And what we found, and we published that last year, is that there was significant gene amplification the moment you start to select mutants in increasing TCP concentrations. So you isolate your resistant strains, and then you just do the sequencing. And what we show here is that the BLATM1 gene in this element was amplified 100 times or 10 times when you go from wild type to the ones that were selected in 8 microgram per mil of TCP during these experiments. And we looked at that, and in the end this implies that a quarter of the bacterial genome is used to, let's say, sustain or help sustain these elements that battle TCP. So this is very invasive for bacteria. It was said to be a new resistance mechanism, although, let's say, amplification of resistance genes has been documented before, but not with a 10 kb element around it. So, but anyway, we have the manpower and the technology available to bring it till here. And this nicely brings me to the last part of the talk. That's genomics. What is the role of genomics in rapid antimicrobial resistance testing? Well, rapidity is already there. Using the Minion, the Oxford Nanopore Minion tool, there's a little thing, I'm sure you have it in the lab here as well, that will give you gazillions of sequence reads in relatively short time. And for doing a crude bacterial genome, if you have sufficient DNA available in your extracts, then an hour's time of sequence accumulation may suffice to get you a nicely annotatable, analyzable genome. And this for microbiology covers 3 domains. So the impact of this technology is potentially huge, and it will affect the detection of pathogens from clinical materials. Already briefly discussed that. Epidemiological typing at the bacterial whole genome level. If you have an outbreak in your clinical setting, then by looking at the accumulating of SNPs over time, you can precisely trace where a bug came from and how it traveled through your patient population over time. And then the other thing is translating genotypes into phenotypes if you have the right databases available. I think I already briefly addressed this point. Can this be done at reasonable cost, at high speed? with good reproducibility and sensitivity and specificity. That remains to be seen, but for some of the current applications, that is already the case. So doing bacterial epidemiology with this type of technology can be done in real time. There's a little spin-off company from the Sanger Institute in Cambridge. If you bring your strain in the evening, you'll get a dendrogram in the morning. So that's where we are already with this type of technology. Then you have to have cultured the strain, so that part of the diagnostic procedure is not taken into account. But with a colony on a plate, you can do a whole genome analysis sufficiently dense and a bioinformatic analysis afterwards that would allow you to compare strains at the SNP level. And where you can compare the data that you generated last year with the data that you generated today or tomorrow. But this is going to be something that we really need to look into. Also in one of the more recent JCMs, there was a paper on Staph aureus sequencing within a day shift. Again, with limited and, let's say, simplified DNA isolation, with limited and simplified library, sequence library construction, with not like a full coverage of the genome, but with a sequence analysis that suffices for doing very reliable strain-to-strain comparisons. So this is already there, and the only thing that you need for that, in addition to the sequencing technology and your rapid clinical processing of isolates, is a database. And we've been looking at the database, let's say at sequence quality in the public domain. And again, a little warning here, you have to be careful with what you look at. If you just believe anything that's being published today, then you're fooling yourself. We found out that about, let's say 50 to 20% of the genomes that are in the public domain do not meet the standard Illumina quality criteria, for instance. So if you start developing databases, and again, for doing bug identification, epidemiology, and also AST, this is absolutely key, then you have to set your standards very high. So looking at Genome, we have a resistance database that contains about 10,000 different markers right now. That goes from gene presence and absence to point mutations, to deletions, insertions, and even some things that have to do with promoter polymorphisms and transcriptome changes. And if you have this type of databases, then the only thing you have to do the moment you have a new sequence is to run the sequence through the database and see whether you get hits with the known markers. Again, don't forget that in the current world, there's a steady increase, a steady discovery of new resistance markers. So this is always a dynamic system that should be maintained and updated on a regular basis for the emergence of new resistances. And then if you do, let's say, country-wide studies like we did in Bangladesh with Salmonella typhi, you can read about it in last year's MBio paper by Tanmoy et al. Then you'll find new resistance markers that you find through criminal resistance in new GRA mutations and stuff like that. So these studies are absolutely key to not only define what's happening in the country on a resistome scale, looking at all the resistance genes, but it will also help you expand your database. And then sort of the last point I want to make, if you have an organism like Salmonella typhi and you know the phenotypes, you know the resistance profiles, then translating a genome sequence into a phenotype is a very reliable process. We get sensitivities and specificities here that are better than what we do with 5K2. So again, in this case, Salmonella typhi, a relatively clonal, well-conserved, not too dynamic at the genetic level organism, can be, let's say, can be analyzed with respect to its phenotypic resistance at the genome level, which is what has been demonstrated for TB as well, for Staph aureus, the work by Derek Crook and his colleagues in Oxford, from which they transformed the entire public health system in the UK, the entire public health system in the UK, from a phenotypic tuberculosis AST system to something that is now exclusively genomic is another example of this type of sensitivities and specificities. Again, they need to keep track of the emergence of new mutations. They need to look at that in great detail with great attention. But they even showed that this genomic approach, where you have to sequence the strains and where you have to look at bioinformatics, where you have to perform bioinformatic analysis, is less expensive than having a technician doing phenotypic AST testing in a security laboratory for 3 weeks with all the time lost but spent and the biomaterials that you need. So this is really something that sort of sustains the model that the same people already sort of promoted in 2012, where you have samples and let's say methods, pre-analytical methods. And again, these do not exist today still. This is one of the areas that was not developed, that did not develop as extensively as the omics arena. But if you have methods that would help you purify DNA from all of these different samples with high efficiency, then you sequence, you read and process and assembly, and then your database of new sequences allow you to species ID your bug, to look at relatedness at the SNP level or at the MLST level, whole genome MLST level. With the resistance and virulence knowledge base, you can identify, detect the most problematic or potentially problematic bugs. And using your own data and your own databases and communicate with people in different hospitals and in different countries, you can correlate what you find in your own hospital with what's happening on a global scale. And whether this is going to happen, I don't know. I think certainly in part. The Illumina iSeq is a machine that would fit on this part of the stairs. It is in a significant number of Dutch labs available already. I don't know the French situation, and people are communicating with each other using these genomic data in order to do infection prevention and to help establish a sort of a national surveillance center on emerging problems with virulent bugs or with resistant bugs. This is where I would like to stop. The only thing that I would like to draw your attention to is that we published a little paper last year that also describes all of the issues around the development of diagnostic tests. And I should not recommend because I'm among the authors. But today I would really like to recommend that because it gives you a nice sort of supplementary view beyond all of the science that is involved in this. Science, absolutely key. New developments, new strategies, new technologies. You have to have a scientific mind to put it all together. But in the end, as clinical microbiologist or infectious disease specialist, you should also keep in mind that what we discussed about scientifically speaking, stops here, where things become a boring project at an industry, where you need to look at technical feasibility, risk analysis, where you have to draft all sorts of silly dossiers, where you have to look at what the competitor is doing. All sort of stuff that can frustrate the translation of a promising technology into something that is in the end going to serve the patients. So this paper sort of summarizes all of that, and it may help you to either educate yourself a little bit in that or to help you generate your own opinion on all of it.
It's your taxi.
Yeah, customers. Customers, I think, yeah. So this is it. These are some of the people who are involved in all this project at Untitled, whom I would very much like to acknowledge. And not all of the work that we've been doing with these people has been mentioned today. Thank you for your attention.